Welcome to today's video on experimental method of data collection. In any research investigation, the first step is collection of data from a sample or from a population. Though there are other methods of data collection like survey method, observation method, etc., the most important method in life sciences is experimental method of data collection. Since the topic is vast, we have divided it into sections. Experiments in life sciences may be broadly classified as in vitro, in vivo, and in silico. In vitro is the Latin term for within glass, and it refers to experiments that are carried out in a controlled environment outside a living organism, like in a test tube or a petri dish. In vivo is the Latin term for within living, and it refers to experiments that are performed on whole living organisms like a human subject, laboratory animal, or a plant. In silico refers to experiments that are performed in a computer or via computer simulation like structure-based drug designing, protein structure prediction, etc. In silico, studies are usually referred to as dry lab experiments, and in vitro and in vivo are wet lab experiments. Examples of in vitro studies are experiments with cells derived from multicellular organisms. It may be cell lines like normal or cancerous cell lines, or it may be tissue culture from explants. Experiments with subcellular components like isolated mitochondria, etc. Experiments with cellular extract or extracts from organelles. Experiments with purified biomolecules like nucleic acids, proteins, and experiments with bacterial cell cultures for antibiotic sensitivity testing, etc. The advantage with in vitro studies is that it can be repeated many times, and inference will be straightforward because we don't encounter all the confounding variables that we may see in whole organism studies. But the results are inconclusive and they have to be validated by in vivo studies. Examples of in vivo studies are with whole plants, effect of different type of fertilizers on plant growth, composition, etc. For whole animals, it may be toxicity studies or studies with investigational new drug prior to clinical trials, and experiments with human subjects may be clinical trials for drugs, diagnostic and therapeutic procedures. In vivo studies yield conclusive results, but we may face a constraint of sample size. We will focus on data collection by in vitro and in vivo studies and steps that have to be adopted to reduce the error. Errors can arise during sampling itself and they are referred to as sampling errors. And as a result of sampling error, the sample that we get may not be a true representative of the population. So the results obtained for the sample may not be extrapolated to the entire population. It may arise both in random and non-random sampling. In random sampling, it arises by chance because even two samples that are drawn from the same population may not yield the same type of data. And we can reduce this type of error by increasing the sample size. In non-random sampling, it results because the criteria that have been laid down, the exclusion and inclusion criteria, have not been successful in drawing a good sample from the population. This type of error has to be reduced by unbiased, stringent criteria to ensure that the sample is representative of the population. Non-sampling errors arise after the process of sampling is complete. It may arise at the planning stage when the experiment is being designed or when the experiment is performed, that is the fieldwork or benchwork stage, during the tabulation stage or during the computation stage when the statistical analysis is performed. Non-sampling errors may arise whether we work with a population or with a sample because it comes only when the experiment is designed and conducted. Tabulation errors have to be avoided only by repeated cross-checking to ensure that the correct raw data has been chosen to calculate the central tendencies and measures of dispersion, and that the correct mean and central tendency posted against the parameters, etc. And statistical analysis, we have to ensure that we choose the correct statistical test for a particular data, and ensure that the data is satisfying the criteria required for the particular statistical test. Before we move on, let us understand the terms control and treatment groups. The term treatment, we should not take it in its literal sense. Treatment is any independent variable which the experimenter manipulates. So suppose we are doing a study to understand the effect of different pollutants on plant growth. We will be exposing the plants to different pollutants, and that is manipulated by the experimenter. If they are working with a lot of pollutants, then we may expose the plant to different type of pollutants, or with a single pollutant we may expose it to different concentrations. So these are manipulated by the experimenter, and this is the independent variable which we are manipulating. And the dependent variable is the plant growth. So because we are exposing it to different pollutants, we want to see how plant growth is getting affected. So it becomes the dependent variable. The groups that are receiving this treatment are referred to as treatment groups. But whenever an experiment is designed, we should definitely have a control group. So for the above experiment, the control group is a group that is not exposed to any pollutant. The advantage of having this control group is that we become sure that the dependent variable change which we are observing is attributed only to the independent variable, because there may be other variables which are beyond the control of the experimenter, but the control and test groups are exposed to similar type of variables. So any change that we note between the control and treatment groups should be because of the independent variable alone. According to R.A. Fisher, for a good experimental design, three important criteria have to be kept in mind. First is replication, next randomization, and next local control. They act complementary to each other to increase the accuracy and reduce the error. Replication, as the name itself implies, repeating the experiment many times. Randomization means you are randomly allotting the experimental units to the control and experimental groups. Local control means we are trying to ensure that the experimental units that are allotted to the control and treatment groups are as homogeneous as possible, so that for the difference between the control and treatment groups will be only due to the different treatments that are employed and not because of any differences in the experimental units. So, there are many extraneous factors like genetic variability, environmental factors, etc., which are beyond the control of the experimenter. But if we do the replication of the experiment, and if we have also incorporated randomization, that means randomly allotting the experimental units to control and treatment groups, we will be successful in steadily decreasing the error. And decrease in standard error is proportional to the square root of r. That means the more number of times you repeat the experiment, we'll be able to reduce the standard error, which is standard deviation by root n. Precision of an experiment also increases by doing replication, but we cannot be continuously replicating an experiment because it involves both cost and time, but we have to generally ensure that R is not less than 4. Now, In vitro experiments, as we have already seen, you can replicate it many times, as many times as required. It is easy to do it. But even in vivo experiments, we have to ensure that the experiment is repeated many times by increasing the sample size. If we are working with two groups, maintaining a good sample size may be easy. But even if we are working with many treatment groups, you have suppose one control group and many treatment groups, we have to ensure that we have a minimum of 5 or 6 animals or plants in each treatment group. We will focus on randomization and local controls in the subsequent videos. Thank you.